Hi everyone, and welcome to today's webinar, which is on tools and partnerships for sustainable increases in rice production. I'm Dusty Vyas and I'm a senior scientist at LGC Biosearch Technologies, and I'm going to be the moderator for today's event. Today's educational webinar is presented by LabRoots and brought to you by LGC Biosearch. And if you want to know more about us, go and look at our biosearchtech.com website. And we would encourage you to participate by submitting the questions that you may have during the presentation. All you need to do is simply type them in the Ask a Question box, which is on the right-hand side, and then just press click. We'll answer as many questions as we have time for at the end of the presentation. You may also ask a question in the box, and if you are having any trouble seeing or hearing the presentations, let us know through there. I'd like to now welcome our speaker today for today's webinar. Her name is Dr. Catherine Steele, and she's a senior lecturer in the Sustainable Crop Production at Bangor University. And for me, she's very, very important, and she's made a significant contribution to rice market development globally. For a complete biography of our speaker and myself, please look at the biography tab on the top of your screen. So Catherine, please feel free to begin your presentation.
Hello, this presentation is about tools and partnerships for sustainably increasing rice production. So the tools are a new database of off-the-shelf markers that can be used for this, and the partnerships that we developed were between rice scientists, biotechnologists and bioinformaticians. So molecular markers have evolved over the last few decades. The first generation you may be familiar with were mainly hybridization based or random PCR markers. The second generation were more sophisticated random markers such as AFLPs, and then directed PCR markers targeting simple sequence repeats. Now, a lot of rice breeders will be very familiar with these microsatellites or SSRs because they've become very well applied in rice breeding mapping and selection using marker-assisted backcrossing. That's been used for well over 20 years now by breeders. But more and more sequencing, first by Sanger sequencing, and then the advance of next generation sequencing has increased the number of single nucleotide polymorphisms that we call SNPs that can be identified. And these are much more widely scattered across the genome of rice and much more likely to be within genes or very closely linked to genes rather as compared to the microsatellites, SSRs. Since about 10 or 15 years ago, the cost of next generation sequencing has reduced, and this has enabled a number of next generation marker systems such as RADseq and GBS, which are very similar techniques, and now low coverage whole genome sequencing. But also, just because so many more genomes of different varieties have been sequenced and become available, there is a huge amount of information now on single nucleotide polymorphisms that rice breeders can tap into. So we built our project around rice breeders, so 4 different organisations represented on this slide, who wanted to all improve their marker-assisted breeding using SNPs. And this consortium was set up through a funding initiative from the UK government known as Innovate UK. And we worked together with Biosearch Technologies, as well, or LGC, as I'll probably call them in this talk, to develop better methods for use by those breeders. So it was a partnership with the breeders' aims in mind, to make sure that what we were developing would actually work and not just be what a bioinformatician thought we should use. So we were targeting Asian rice breeding, so mainly indica rice, but we were making sure that we were incorporating a greater diversity than just indica in our design of markers. So the partners, the first in Nepal is Anmolbu Private Limited. Anmolbu means beautiful, sorry, it means precious seed, I should say. And they are a small breeding company. They do not have their own breeding station. They grow all their test varieties with farmers on farm, and this works brilliantly. Also in Nepal, on the top right, we have NARC, and we were working with two divisions of NARC, the National Agricultural Research Council of Nepal, and they have breeding programs focused on cold tolerance, blast resistance, and bacteria leaf blight, which I will just call BLB resistance. The third partner going round in a clockwise manner is Nibji in Pakistan, and they are looking to improve aromatic rice for salinity and drought tolerance, and also BLB resistance. And then on the last section, we've got Skuast from Kashmir, and they are also interested in improving aromatic rice, and their focus in this project was to look for resistance markers to Bacane disease, which is becoming more prevalent in rice growing regions of Asia. So producing sufficient rice for Asia involves not only targeting yield, disease resistance, and grain traits, but traits for climate adaptation, such as heat stress, drought tolerance, salinity tolerance. So it's a huge number of traits for breeders to be able to grapple with. And molecular breeding has tended to focus on simple traits such as disease resistance, where we can identify single genes associated with disease resistance and use them in marker-assisted selection using SSRs. But when you're wanting to incorporate multiple traits, then a more sophisticated technique for screening and selection and a wider number of markers is required. We also need to consider crosses with locally adapted land races that are bringing those new and novel traits that may improve climate adaptation in rice into higher yielding varieties or varieties with specific quality traits. So using these more efficient tools and approaches can hopefully improve the efficiency of breeding rice. And by working with partnerships and sharing resources, sharing best practice and sharing experiences and being able to help each other to solve problems going forwards are really, really important. And that's what we managed to do over the 5 years or more that we've worked together in this partnership. So the location of the partners is shown in this map. I'd just like to show this to sort of show, although we're working in 3 different countries, Pakistan, India and Nepal, the area where the rice is being produced is quite similar. This whole region depends on rice. In Nepal, it accounts for just over half of all agricultural production, 51.7%. And there's a huge amount of rice genetic diversity across these regions where we're working. So in Nepal, the work was done in a number of regions. So the yellow part is the terai, where rice is grown in flooded fields. And then in the green area, you've got the mid hills where rice tends to be grown in more upland sloping conditions. And NARC themselves are based in Kathmandu. So you can see the arrow in the middle there in the central region of Nepal. But NARC also have a research centre for doing their screening down in the Terai in Hardanath. And then APL have 3 different sites on this map, all in the Terai, but spread out across Nepal. And this gives a good amount of information for screening for disease resistance across the whole region, or the whole of Nepal. So the project initially started off doing next generation sequencing of 9 different rice varieties. Now, this diagram on the right shows the 12 chromosomes of rice. Each block is 12 chromosomes, but each block is broken down into 9 separate columns, and the diversity of, or the density, I should say, of SNPs is shown in the red colour. So where the red is darker, there is a greater diversity of SNPs that are potentially useful for developing into CASP technology. We used this whole genome sequencing to compare varieties, these 9 varieties that had been selected by our partner breeders. And they were varieties that were all being used as parents in their breeding programs. A lot of them were donor parents of disease resistance and other important traits. So from that, we found 300,000 SNPs and 39,000 indel variants, and those were filtered through bioinformatics to make sure that they would work as CASP designs, so that they could be assayed using that technique. And those have been published in the paper that's cited at the bottom here. And along with the coding, which is available to be downloaded as a supplementary information, that you could, if you've got your own rice genomes, you could follow that to try and find CASP that may be specific to your populations, if you can't find them from these off-the-shelf ones. But hopefully what I'm going to go through for the rest of this talk will inspire you that we have now got off-the-shelf markers that can pretty much be used in any rice breeding program. So a little bit more detail about these tools. CASP is a technology that is licensed to LGC Biosearch Technologies, and it is a PCR-based technique that is almost as flexible as microsatellites. It can be used in small numbers to do exactly the same jobs that you would use microsatellites for in marker-assisted backcrossing and QTL mapping. LGC have a second method, SeqSNP, which they developed during this program. And we were very lucky at the end to be able to include some SeqSNP in the research with 2 of the partners. And I'll come on to that. But SeqSNP is a high throughput screen of thousands of markers. Now, CASP and SeqSNP show concordance between the technologies, and we have demonstrated that by designing SeqSNP, well, I should get this right, by designing CASP at a large scale and then using those marker designs for CASP for SeqSNP, we can allow the 2 to be the same loci can be tested with both techniques, but it only works because of the bioinformatics that we used on the CASP designs. And the beauty of this is that the service is provided as a full service, including DNA extractions, and you don't actually need to run these in your own lab if you don't want to. You can order CASP to run in your own lab from LGC, but you can, and we were expecting our partners to want to do that because they'd been doing that with microsatellites. But when they realized how simple it was to just order the CASP through this full service with LGC and just send their leaf samples, and you can see on the bottom right-hand picture there, the team in Nepal sampling leaves to send off in these boxes to LGC. When they realized how efficient that was, they haven't actually bothered to run the CASP in their own labs. But clearly CASP can be run in your own labs if that suits your program. So we actually improved on the original designs from the 9 genotypes in the second tranche of funding that we had from the UK government. And that allowed us to incorporate 121 additional genomes to take account of extra variation. And then we were able to improve the designs of the CASP assays so that they looked at non-target SNPs or indels in the sequences of all of these additional diverse genomes from indica and japonica and varieties from many different countries, so that they should work in a much wider set of genomes. And from those, we were able to home in on 1.3 million improved assay designs, of which nearly 100 million were predicted to be functional. That means they're in genes and have an effect on the protein product that's produced from the prediction of the sequences. And between them, between any two of those 129 genomes, there were a minimum of 23,000 pairwise polymorphisms that could be found. So even very closely related varieties, you could find useful polymorphisms among them. So from those, the project allowed us to validate a set of 5,000 that we selected from that 1.3 million based on the advice of our breeding partners. And the success rate of those becoming usable wet lab validated CASP was between 72 to 90%, depending on a number of factors. So out of those, we have got 4,867. The number's going up all the time whenever we test a few more. And so that on the right there, you can see 3,355 of those used on 178 genotypes, of which only 10, I think, were used in the original sequencing. So the rest, over 160 genotypes, varieties that hadn't been tested, hadn't been used to design these primers. They were tested blind, as it were, with these markers, with these CASP markers. And the results there show that they can discriminate the japonica varieties, which are on the bottom left in purple, from the indicas, which are on the right in the light green colour. And all the basmati varieties are in the red colour. And then there were intermediate varieties. You won't be able to read the names of those, but for example, Pusa Basmati 1, although it is a basmati, it comes in that intermediate set because it contains diversity because it's a modern variety and it's had introgressions from some of those other groups. So modern varieties, when they've been had that complex pedigree, they tend to fall into that intermediate category. And so our CASP set were very good at distinguishing these different types of variety. So the success rate was, as I say, 90% for CASP. We also validated, we tested 15,000 of the designs in SeekSNP, and the success rate of those for design to wet lab validated was 93%. More on SeekSNP in a bit. And all the loci that we've designed, so all designs are going to be available very soon from LGC Biosearch Technologies in a searchable database. And from that, you will be able to look for your genome position of interest, or a gene that the idea of a gene that you are interested in and the regions flanking it. Or even you can look up microsatellite markers and SNP markers that have been used in chips by IRI. So you can go into that database and look at those regions, and that will tell you which novel CASP designs are in those regions. And then you should be able to click on them and download the information that you can then use to order if you want to try them in your own breeding program. And again, it's great if you send your leaf samples, and then LGC can just send you a spreadsheet of your genotypes at the end. And I think it's just going to be an amazing set of new off-the-shelf markers that should work in. Well, yeah, you'll probably get 1 in 9 will give a result. If you choose a batch, 1 in 9 should give a result. And if they're polymorphic in your material, then you should be able to use it and work with it. So the next few slides are going to go into a bit more detail of what our partners have achieved using these new markers during this development process and more recently. So APL in Nepal are very well, in Nepal, aromatic rice is very important. It increases farm income by about 40% if they can sell a fragrant aromatic rice. And prior to this project, Sonola Suganda had been bred by APL as the first modern aromatic rice variety. It was released in 2014, and it has Pusa Basmati 1 as one of its parents. So you can see that on the top right in the little photograph there. That was crossed with 2 local land races. One of those, Gadjali Machino, is shown on the photograph. And from the progeny, APL used CASP to identify lines that carried the target traits for aroma and for other traits such as disease resistance, and also selecting the local variety recurrent parent for background traits that would bring that adaptation to the local environment for Nepal. And one of the markers is targeted to the fragrance allele, the BADH 2.1 locus of basmati rice. Well, it's in a lot more fragrant rice than just basmati. But there are also minor QTLs for aroma as well. So we've got, in our set of markers, we've got CASP that will target those as well. And so this breeding program really advanced using CASP. I think at least 3 generations of marker-assisted backcrossing and selection was done by APL. Remember, they don't have a breeding farm, and they don't have their own research station, and they don't have a lab. So they were doing the selection in farmers' fields, and they were sending the leaves off to LGC for genotyping. And we were working with them to help interpret and smooth out the process for them. And they've got really good at doing it now. And in addition to that, right at the end, we were able to run 15,000 seq SNP. And the aim of that was to actually look at pure line selection. As I mentioned, APL are a seed company. They sell certified seed to farmers in Nepal, and they need to make sure that seed's pure. We were able to extra purify Sonola Saganda through the use of seq SNP. So the next slide shows, on the right, you can see all of the Sonola Saganda samples that they tested with seq SNP, and within that population, out of the, so we ordered 15,000 seq SNP, of which 13,115, I think, were worked and were polymorphic. And out of those, in just the Sonola Saganda population, there were 1,207 that were polymorphic. And you can see in this principal component analysis that the true type are the ones at the bottom left, and you could see 3 clusters where there's some off types within that population. So the breeders in APL were able to discard the off types from that and make sure that their certified seed was really pure for Sonola Saganda. So that was just from the original run of the seq SNP. But it wasn't just Sonola Saganda that was included in that. And the next slide will show those. But I'm just going to mention while I'm on this slide that another organisation, NIBG in Pakistan, were also able to benefit from running seq SNP. And so we're really pleased that both organisations were able to do this. So within the NIBG budget, we had enough funds for 2,000 seq SNP to be tested, and they were using it in a mapping population between Super Basmati and an IRI donor variety. And these, there were 1,291 that were polymorphic within that cross. And again, these were the seq SNP designs were from the full set, and we hadn't used Super Basmati within that to design the. We hadn't used either of these parents actually to design or to sequence for the design. So it was a good success rate of polymorphism in the NIBG set as well. So APL, back to APL, their entire breeding program, they made 11 crosses during our project, targeting, as I say, the disease resistance and aroma. And they also did linkage mapping to confirm that some of the bacteria blight resistance genes were associated with the Cas markers. So previously they'd relied on microsatellites, and these Cas markers for this designed for the same targets were equally good at co-locating in the breeding program through the field trials where they screened them for BLB, bacterial leaf blight resistance. So they multiplied those new BLB resistant lines, and those are in the pipeline for resistance. In addition, the aromatic crosses from the 2 crosses mentioned in the previous slide, they've now got 10 advanced aromatic lines in yield trials in Nepal. And in addition, from a cross between Hardanath 1 and another donor variety, they have developed super early lines. So these were selected for early flowering. And this means that and they're also highly disease resistant. And this means that they can be grown early in the Chaiti season. They're very early sown, and they allow farmers more flexibility and have greater income through the season. So that's APL. NARC, National Research Programme, all of the seed certification and release is done through the National Rice Research Programme, NRRP, and so they lead the coordinated field trials. And NRRP collaborated to evaluate the crosses from APL in their field trials as well for Blast, BLB and Aroma. And they're now evaluating 33 advanced entries under multi-location trials. And this is ongoing, and hopefully varieties will be released over the next few years. So going back to the seek SNP, the 15,000 seek SNP that were originally ordered were tested on 108 genotypes. And these are shown in this PCA on this slide. So advanced breeding lines from APL are in the red. Sorry, advanced modern varieties are in the red. Advanced breeding lines are in the green and light blue at the bottom. So the two best aromatic breeding lines are in the light green and the light blue, and the Sonola Saganda is in the darker blue, slightly darker blue. So what we did for seek SNP evaluation, we found that 67% were highly informative and polymorphic within these 108 genotypes. And remember, these are quite non-genetically diverse. They're quite uniform because they're all from the breeding program of APL, but apart from the ones in red. And they are these markers are giving a huge amount of discrimination between them and really useful for selecting and also selecting against the off types. So when the analysis that was done was principal component analysis, and what my colleague John Whitcomb did, who did this analysis, was to take the original 13,115 polymorphic SNPs and run it in the analysis, and then he just took out the ones that were polymorphic in Sonola Saganda to run that analysis, and then he was able to look at the individual breeding lines and run the analysis again with a smaller subset of markers, each time repeating the principal component analysis, and then any markers that gave exactly the same pattern of allele calls between the same lines, he would just replace it with one of those to reduce it to a smaller subset that could still separate in principal component analysis those varieties. And that led to 39 highly informative SNPs, which of those we then ordered, or APL then ordered CASP to be done on those using using the lines from their fields, and that showed that 67% of those seek SNP were suitable for running as CASP. And if you remember that this is a non-diverse set of lines, the ones that were monomorphic in this could if you added those ones in, you'd get an even higher success rate. But because they were monomorphic in this population, they weren't usable, but they may have been usable in a wider diverse set. So yeah, John Whitcomb basically says conversion of CASP from SeqSNP using these designs is brilliant, and he's so thrilled with the way that the APL have been able to improve their seed purity and improve the purity of the new breeding lines as well. So they're taking these CASP forwards for subsequent selection in their marker-assisted back cross breeding program. There we go. So moving on to NARC, the second division of NARC, which is biotechnology. So this has this was run by Resham Angai, and he was focusing on improving high altitude rice and also looking for disease resistance and agronomic traits. So there were two varieties, Cumul 4 and Jumli Marshy, which were crossed with donor varieties. And these were some of the lines that were chosen for the original sequencing. And from this, 18 blast resistance enhanced rice lines were selected. Resham also did QTL analysis in the population and confirmed the CASP for the blast resistance gene PI33 was co-segregating with blast resistance. And this was in high yielding cold tolerance breeding lines. And he also looked at a diverse set of lines that he was interested in using as parents in breeding, and has identified traits, markers, CASP markers associated with traits for agronomic, photosynthetic, and biotic and abiotic stress tolerance traits. So moving on now to Nibji in Pakistan. So Nibji have, as I mentioned previously, also been looking to improve aromatic rice, and their main focus has been to improve super basmati for disease resistance, so bacteria blight resistance, and they've got the success in producing a single genotype with 3 resistance genes pyramided into it at 90% homozygosity. They've also confirmed that I think they've got 5 CASP markers for XA5. It's like a haplotype, and when these are all present, they've got homozygosity within their lines. So they're very confident with those XA5 markers, but also they've been using the others, and they're getting really good results. So they are expecting one of those disease resistance basmati types to be released by 2026, and they're very confident about that. They've also selected numerous lines with combinations of 2 XA genes for bacteria blight, and they've been screening a training population for genomic selection as well, to look for to look at the diversity and try and associate those important traits for breeding with markers, and then use that going forwards in their breeding program. And finally, they've got another cross with super basmati and another IRI line, which is a drought tolerant line, that they have identified QTLs for drought tolerance and grain traits. And the graph on this, that slide is showing some of their QTLs on chromosome 3 that they have identified. And that is being developed by the team at Nibji into a publication which will be coming out hopefully fairly soon. And then moving to India. So SCUAST, I think is how you're supposed to pronounce it. They are a university in India they're also interested in aromatic rice. But what they used, what they did when they were able to test a large number of casp, was to screen a diverse collection of accessions from India, from the Himalaya region of India. And they have looked at that and identified, they've run cluster analysis and identified 6 casp that are largely differentiating local land races from the temperate exotic germplasm. So those casp are particularly useful for maintaining local adaptation going forwards. And that's already been published in Genetic Resources and Crop Evolution. SCOAST also have a mapping population, they've developed a mapping population for Bacane disease. And they've been screening lines for resistance, and you can see some of their resistant and susceptible lines clearly in this plug tray experiment. And success has come. They've identified QTLs, a number of QTLs for Bacane resistance, and 2 particularly interesting ones, one on chromosome 2 shown in the diagram here, and one on chromosome 6, using QTL mapping based on the CASP genotypes that they ordered through the genotyping service of LGC. And they're currently continuing to do that with fine mapping, using those markers to home in on hopefully the candidate gene. And again, a paper is in preparation and should be released, should be published soon, about the Bacane resistance gene in that population. Resistance QTLs, I should say. So it's been an amazing few years, 5 or 6 years at least. We've done a huge amount and worked with a huge number of rice breeders, and they're also pleased with what we've achieved through the development of CASP and then going on to the seek SNP. The project has been highly successful, and we've summarised here some of the achievements. We have 1.3 million new rice genome assays that have been designed and are going to be made publicly available off the shelf, so any breeder can order them and run them themselves. We have assayed over 8,000 rice accessions in this project across all the partners with either CASP or seek SNP. And we've got at least 4,867 CASP that have been validated across the entire project. That number is going up every few months when we run another project. We've just run a project through Bangor with Pakistan researchers targeting CASP in amylose genes that hadn't been run and validated previously. So that adds another 10 or 12, I think, to our set. We've got Nepalese scientists who've been trained in QTL analysis. I went out and did a training course during the project so that they can use the data more effectively when it comes through. We've got at least 26 different rice collections or populations that have been genotyped in the project. So that's for diversity, genetic diversity screens. We've had 12 crosses that were made specifically for this project. We've run 10 rounds of CASP. That, again, that number's going up. But on this project, there were 10 rounds that LGC has contributed to the project. 8 early careers at least have had their, 8 early career researchers have at least had their careers enhanced through the project activities. And so far, there are 3 published papers in international journals. That's the Indian Himalaya land races, the original CASP designs, and the location of markers associated with disease resistance genes as well. That is published in agronomy. There's going to be at least 2 more papers coming out, and then we are in the process of publishing the full CASP design paper that will accompany the database release as well. But then we haven't released any new varieties yet from the project, but as you will have seen going through, there are a number of new high-yielding varieties that will be that are in the pipeline, and pending their performance in the all area trials, they should be expected to be released in the coming years in Nepal, India and Pakistan. So it is a case of watching out for those and keeping in touch with the developments going forwards. So I would just like to finish with the acknowledgements here for all the people and organisations who've been involved with this work. It's been a really amazing team to work with, and a lot of people have put a lot into this. So we've got the team from Bangor, past and present colleagues, the SCUAST team led by Arif Shikari, the NIBGI team led by Dr. Mohammed Arif, and the APL team, they're all great, and NARC led by Resham. And we had a project partner in the Agri Epicenter that helped with the funding reports to the funders, and then the LG team and Dusty as well, who is part of this meeting, who's been a real support throughout the whole thing and answered all sorts of questions about CASP designs and how to get the samples into the leaf boxes and all sorts of questions that we've all had. And the whole team at LGC have been amazing. And then the funding has come through mainly Innovate UK, but also other UK government sources. And there was some funding to the Nepalese and Pakistan partners from their governments as well that has added to the whole achievement. So I shall stop there. Thank you.
I don't know whether you can see me, but it doesn't really matter. Can everybody hear me okay? Yeah, perfect. Okay, I'm here. So thank you so much, Catherine, for an amazing presentation, which was incredibly informative. So what we're going to do now, we're going to go to the live Q&A session of the webinar. If you have any questions that you'd like to ask, please do so now. All you need to do is type your question in Ask a Question box and submit. And we'll answer as many questions as we have time for. Oh, and they're coming in already. So let's go with the first question. So I've got a question about what source did you take from Nepal for the aromatic rice? So do you know the variety?
So that was actually Pusa Basmati 1. So it was a line of Pusa Basmati 1 that was being grown in Nepal, and that was it was crossed and used for selection, and that ultimately ended up with the development of Sonoda Saganda, which was then the first modern aromatic rice that was available in Nepal. The breeding details are in the papers by Professor John Whitcomb and the colleagues in Nepal, if you want to have a look at them. Oh, what's his name? I can't remember the name of the guy. It'll come back to me.
Perfect.
Krishna Joshi. He now works for IRI.
So thank you very much, Catherine. And I'm sure everybody's going to be looking for these super aromatic varieties now. So I've got another question that is saying that this is a stunning list of achievements. So that's great. So how long since the beginning of the project? How long has it been going on for in total?
Oh gosh, Dusty, how long was it since we had that first conversation when I said to you, can we convert SSRs into CASP? And you were saying, oh, well, it's not that simple, which was great.
Yeah,
So that would have been, go on.
I think it was 2013, wasn't it? It was very early on.
Yeah, I was going to say, I think we wrote the first project proposal in 2014 and we got it funded in around 2015. It might have been 2014. And then, so we had a 1.5 years a feasibility project. And then I think 2017, we started the industrial project, and that ran through to the very start of 2021. We hoped we were going to have 6 months more funding. Unfortunately, due to various political reasons, we didn't. But yeah, we managed to achieve all of this. This is why the database has been a bit delayed, because that end of the funding slowed things down, but we're still working on it essentially. So yeah,
But I mean,
7 years.
But considering, I mean, yeah, 6, 7 years, but the amount of work that was done, and the achievements, and getting the lines into an advanced state is absolutely amazing. So, okay, let's go on to the next question. Do the partners in the project think that the application of CAS markers for marker-assisted selection made a difference to them?
Definitely. I think, well, first of all, APL weren't using marker-assisted selection before this project, so having access to CASP was a lifeline for them for using marker-assisted selection. I think all of the others, certainly NARC and NIBG have used them in marker-assisted breeding. I don't think SCUAST have actually used them for selection. They've mainly used them for diversity analysis and mapping. But yes, they've all felt this has made a huge difference to their breeding programs.
And it's great the fact that, if they've not been able to apply markers previously, the setup of the all-inclusive enabled them to do that.
Absolutely, yes.
Right. The markers that were developed for the community, rice breeding community in Nepal, Pakistan, and India. But do you think these markers are going to be relevant to other breeders in other areas of the world?
So yes, for rice, obviously this is a panel of markers specifically for Oryza sativa. So wherever Oryza sativa breeding is of interest, then there will be markers here that will be relevant and useful for you. I think I made a bit of a mistake when I said it was 1 in 9. I meant 9 out of 10 or 8 out of 10 markers from this set should work in most possible crosses. I'm not quite sure. I was a little bit in the flow and didn't get that right, I think. So yeah, it's basically order at least 10 and you'll get at least more than half of them should amplify, should give you a genotype, and hopefully they will give you useful polymorphisms between the lines you're interested in, wherever you're working in the world on rice. If you're working on rice wild relatives, the success rate will be lower. These have been designed specifically within Oryza sativa, but taking into account a huge amount of variation within that species.
But I mean, the density of markers that were identified there could be possibilities. And of course, using the searchable database, that will make it even more feasible, I presume.
Absolutely.
Okay. Oh, okay. Well, the questions are coming in. What was the rationale for selection of marker sets for targeted sequencing? And as a follow-up, how many CAS markers do you think would be needed for routine quality checking on varieties and hybrids? That's a big question.
Yeah.
Shall I say?
Yeah, go on. Sorry.
What was the rationale for the selection of markers set for targeted sequencing? And then
Okay.
let's start with that one first, and we can.
Okay, so the targeted sequencing, so that's the SeqSNP. So basically, we designed, our rationale was to design CASP in this project. And so the SeqSNP were kind of a byproduct of the CASP designs. So the rationale for CASP was a design that was going to work for CASP. So to run a CASP, you need to have either a SNP or an indel that is unique, that doesn't have any extra variation or more complex variation. So it needs to be one that is going to work as a CASP. But we also then when we ran the SeqSNP, we chose markers that we hadn't actually run as CASP previously, to then up the amount of coverage of the genome within the available budget. The beauty of SeqSNP is that you can run a lot more markers than you can run in CASP. And actually, if we were doing the project now that SeqSNP is available, I would start off with running SeqSNP that are designed as CASP, and then use those to inform the set of most useful CASP to zone in on, as it were, because you can run a lot more SeqSNPs for the budget that you can do for CASP. So yeah, it's kind of back to front. We chose the SeqSNP that we hadn't run as CASP, but you could start off using just SeqSNP and then home in on the ones that you want for CASP if you've got the budget to do it that way around.
I mean, in actual fact, you can, there's the SeqSNP is kind of enabling verification, within the genome, and then you can, as you move down the breeding program, you could convert them to CASP to have a lower density set of markers.
Yes, absolutely. So what was the follow-on?
The follow-up was, how many CASP markers do you think you would need for routine quality checking on varieties and hybrids?
Okay, so basically when we did the SeqSNP, we were targeting markers that would be useful for purification. So we were looking within very closely related lines, and ultimately wanted to identify the ones that would be polymorphic. But once we got that down to that, we had it was in the 30s, the number of CASP that we actually needed. Cost-wise, I think the smallest number that it's worthwhile running is either 24 or 48, I think. Obviously, Dusty, you know the pricing structures, and people need to ask what those are and how many. But I think 24 is the smallest number of CASP you can order. If you want to run less than that, it's worth trying to find some extra ones just to fill up your set,
to make it cost efficient.
Yes, yeah,
for sure.
But you can, you can just get away with a small number, depending on what you're actually interested in doing, whether it's marker-assisted selection of a particular locus, for example. I mean, if you just want to, you can just run the fragrance marker. It's just one, but it will tell you whether you've got varieties with that allele or the wrong allele. But yeah, it's not cost effective to just run one CASP.
No, definitely. Well, I mean, the effort it takes to produce all the DNA, then the obvious there's it's better to choose a few more. So this is, we've only got a minute left. So this is a question from me. Do you think the resequencing, the data that was used, part of the resequencing, develop haplotype maps for imputation?
You could. We've not used it for haplotype maps, but the data are all there. The sequences that we did in this project are all available. The first 9 are all available in databases, so you can actually go and find that original sequence data and use that for that analysis, haplotype mapping. Yeah, it's all out there. I think there's 2 extra that we need to load up from the last lot of sequencing that was done for the newer designs. But it's all in the public domain, or will all be in the public domain, and all the other sequences are in the 3K Rice Genome Project. And there's more and more rice genomes becoming available. So yeah, if you wanted to do that, you can do that. Yeah. But we've not done it.
No, but I mean, but that's, that's an idea for people on how they can use the data that's been generated within this. So I think that's all we've got time for. So thank you so much, Catherine. So much fun to do this with you again. And any final comments for our audience? Do you have any final comments for the audience?
Oh, sorry, I was just looking to see if there were any more questions from the audience. But yeah, we haven't got time for that. I think we've covered everything. It's just get in touch with LGC if you want to know any more.
Perfect.
Thanks.
Okay. All right. Thank you again for your time, the time you've taken out to be part of this, and of course your incredibly important research that's going to change people's lives. So I would also like to thank LabRoots for their help with setting up the webinar. Couldn't have done it without them. And of course, LGC for underwriting today's educational webcast. Before I go, obviously, I want to thank everybody who's attended and joined us today, and thanks for the really good questions. And if we haven't answered any questions, then we will get in contact with you and get those questions answered for you.